Hi, it's Hannah here, and today I will be presenting on Parkinson's disease. So, what is Parkinson's disease? It is a neurological disorder with a wide variety of symptoms. So here on the screen, you can see on the left side, we're going to have our motor symptoms, and then on the right side, we have our non-motor symptoms. Now, the important takeaways of these symptoms is the tremors, which are going to be like violent shivers throughout the body, and then our postural instability. So they're not going to be able to stand up without losing their balance, and they're going to have a really hard time walking. Then on the non-motor side of things, they're going to have a lot of mental and behavioral issues. So this can be seen as sleep problems, severe depression, anxiety, and then the onset of dementia in older adults. So Parkinson's disease is classified as a chronic progressive disorder. So this means that not all of these symptoms are going to be present at the onset of the disease, and that they will actually appear and then worsen over time. And this is due to the steady depletion of dopamine that we see in Parkinson's patients. So we're going to see an increasing number of non-functional neurons. So as you can see on the slide, at the top of the screen, we see a normal neuron and a healthy person. So a normal person has a lot of dopamine that is going into the brain, but someone with Parkinson's on the bottom does not have as much dopamine going in to their midbrain. And so these neurons play a significant role in maintaining the cardiovascular, renal, hormonal, and central nervous systems of the body. So if we lose a lot of these normal neuron interactions, we're going to see drastic effects on the body. So for my project, I decided to focus on a form of Parkinson's called monogenic Parkinson's, which is a form of the disease that can be phenotypically present with only a single mutated gene. And out of all of the currently known monogenic Parkinson's disease, they are all autosomal diseases, which means that the genes are only found on the autosomal chromosomes, and they can both be autosomal dominant or recessive. However, 95% of the time, there is no apparent genetic linkage in the appearance of the disease and in the familiar line. So this is what is called sporadic Parkinson's disease. So we're going to see a mutation that takes place in an individual, and this mutation is going to have a biochemical consequence. So it's going to be like a loss of a normal function or a gain of a toxic function. So for my project, I decided to focus on a monogenic form of Parkinson's, which is heterozygous and has autosomal recessive mutations of both the PINK1 and Parkin genes. So the PINK1 and Parkin genes work together on a mitochondrial regulatory pathway. So as we all know, the mitochondria is the powerhouse of this cell. This means that it generates a lot of energy for this cell to use. And you know what needs a lot of energy? Those neurons that aren't working properly. So if our mitochondrial regulatory pathway is not working and we're not creating as much energy as we need, then we're not going to have the functional neurons, and so we're not going to get the appropriate amount of dopamine that we need in order to function properly. So how does this happen? Here on the slide, I have a nice little diagram of a normal, healthy regulatory pathway found in the mitochondria. So the PINK1 gene is responsible for detecting damaged mitochondria. But how does it determine this damaged mitochondria? Well, I have a nice little diagram to explain how this happens. So regardless, the PINK1 gene is imported into the mitochondria. If it is a healthy, functioning mitochondria, like it is on the left side of your screen, the PINK1 gene is going to be cleaved automatically by the MPP and the PARL proteins, which are seen in the yellow and the orange circles on your diagram. Now, if it is a damaged mitochondria, then our PINK1 gene is going to stay in the mitochondrial membrane, so it's not going to get cleaved. In the membrane, it is then going to recruit the Parkin gene to the mitochondria. Now, what does the Parkin gene? Well, when we go back to our big large-scale diagram, you can see in the blue the Parkin recruitment by the PINK1 gene. And what the Parkin gene is responsible for is tagging the defective mitochondria with ubiquitin, which we learned in cell biology will cause the defective organelle to be destroyed in the lysosomes. So, in the grand scheme of things, We want to get rid of the dysfunctional organelles because we don't want to have a damaged, defective organelle using the nutrients and the energy that a healthy organelle could be using to help us with our body. So then what's the problem? Well, the problem is going to occur when the PINK1 gene, or the Parkin gene, becomes mutated and this regulatory pathway no longer functions like it's supposed to. So here we have a nice little diagram of what happens. So on the top, we see what happens with a healthy mitochondria. So if we have a bad mitochondria, it will be removed, plain and simple. But in Parkinson's disease, we're either going to get a lot more mitochondria that are tagged as being damaged. So we're going to remove a lot of healthy mitochondria that could be used to help, or we're going to see not a lot of the damaged mitochondria actually getting out of the cell, so they're going to use the energy that we will need in order to function properly. Now, this seems super complicated, and how the heck did we figure it out? Well, we had to explore the genetic lines and the genetic process in our animal models. So what a lot of researchers did, as is usual in animal research, is to start with mice. So what we did is we looked at mice that were deficient in the PINK1 gene or the Parkin gene, and we expected to see a change in the mitochondrial DNA, which would cause the mitochondrial stress, which would cause the death of our dopamine neurons, blah, 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 except that's not what we saw. The mice actually were able to compensate for whatever happened in the PINK1 and the Parkin gene, and those neurons managed to function normally. So we didn't see any loss of neurons as was expected. However, this wasn't a magic cure-all for the mice because they actually ended up having heart problems due to the oxidative stress that happened when they were compensating for the neuron damage. Instead, the answer to our genetic question was found in our amazing fruit flies. So with our fruit flies, we were able to create a line of flies that were knockout or had the mutant PINK1 and a knockout of the Parkin, and both of those lines that were separate had the same muscle degeneration, the same environmental stress sensitivity, and the same neuron degradation that we would have expected to see. So if they had the same symptoms but they were separate, then this meant that they were acting on the same pathway. So the next step was to then create a line of flies that both had the knockout of PINK1 and the knockout of Parkin. And we saw the same thing, so it's the same pathway. By doing a little more kind of playing around with genetics, we were actually able to see that inserting a functioning Parkin gene into the dual knockout line will save the PINK1 so that the PINK1 gene is upstream of the Parkin gene in the pathway. But why the heck is this important? Why do we need to learn about why the mitochondrial reduction pathway and why the genetics comes into this? Well, there's still a lot of unknowns about Parkinson's disease, and this is weird because it's one of the most prominent human neurological disorders that we see on a clinical basis. So there's still not really a solid hereditary explanation, and we're not seeing a common mode of inheritance in the gene pool. So there's no cure that we can find for this disease. There's no catch-all, and the treatments aren't really working. So by understanding how the disease originates and how the genes mutate and how this all works, we can find clues that researchers can use in order to develop a more effective treatment and prevent it from developing in future generations. Thank you.